Hi everyone at the Coleoptera Society. First of all, thank you so much for the invitation and a special thanks to the Society for supporting this work through the Bell Grant. I'm truly excited and grateful for the opportunity to present here today and to share a project that was made possible in part by your support. In this talk, I will focus on unraveling key evolutionary and biogeographic patterns in one of the most diverse and globally distributed lineages of tortoise beetles, the tribe Cassidini. This grant has been instrumental in advancing our understanding of their systematics and historical biogeography, and I'm happy to show you what we've discovered. So within the diverse family Chrysomelidae, also known as leaf beetles, we have the subfamily Cassidines, which is the second largest subfamily within leaf beetles. Also known as tortoise beetles, they present an incredible diversity with all kinds of colors, shapes, and forms, and also with very unusual morphological adaptations, such as the use of fecal shield as protection from attacks from parasitoids and other predators, cryptic coloration matching host leaves, and also subsociality characterized by parental care. And despite their ecological and evolutionary prominence, the subfamily still lacks a robust molecular phylogeny, a corresponding temporal framework, biogeographic reconstructions, and because of that, the origin and evolution of traits remain largely unclear. So, the tribe Cassidini stands out as the most diverse within the subfamily, including more than 2,000 species described in 87 genera, and is the only tribe distributed across the Old and New World. Despite its relevance, systematic research has been quite limited, and its classification remains a subject of taxonomic debate and systematic uncertainty, primarily due to the heavy reliance on homoplastic morphological characters. And throughout the years, the tribe has been recovered as paraphyletic in multiple morphological and molecular studies, with conflicting relationships with other tribes nested inside the paraphyletic Cassidini. The most comprehensive study on the tribe was by Lopez Perez et al. In 2018, where she did a morphological phylogeny, including 30 genera and 56 species. And in the work, the authors proposed synonym between Aspidi morphini and Cassidini, based on 6 synapomorphies. Well, based on all of that, the goal of the current project is to evaluate the monophily of Cassidini sensu Lopez Perez et al. 2018, infer its relationship with Cassidines, and also biogeographic events and ancestral areas. So for that, we sample 123 species spread throughout 20 tribes of Cassidines. Important to note here that Cassidini sensu Lopez Perez was sampled with counting on 61 genera and 84 species out of the 87 genera recognized for the tribe. We sequenced DNA from museum material, 77 pin-mounted specimens, thanks to the grant, to the Bell Grant offered by the Society. And this data set was complemented with gene bank sequences. The molecular data set was obtained through non-destructive DNA extraction, low-coverage whole genome sequencing, and at the moment we're using mitogenomes. So the assembly and annotation was done using MitoZ and MitoBin. The tree inference was done using maximal likelihood with IQ3, and the divergence time estimation using penalized likelihood with 3PL, and ancestor range reconstruction with biogeobears. So as a result, we have the largest molecular tree of Cassidini, but also of Cassidines, which helped us understanding also the relationships with other tribes. Here, for all taxes, we obtain near complete or complete mitogenomes for material of up to 115 years old. And it's also important to highlight that we recovered high support on most deeper and shallow nodes. Well, it's important also to highlight that the current configuration of Cassidini shows that it really demands a deep taxonomic revision and reclassification. So the Cassidini sensu Lopez Perez et al. 2018 is not corroborated. Besides that, we also recover other groups as non-monophyletic, like Schizorhynchini. And we also recover other tribes as monophyletic, such as Homocerini, Mesonphalini, Goniocenini, Dorinotini, and Basioprenotini. Other finds, we must acknowledge here that based on this tree, there are many results that can be discussed, but given the time, I would just go over a few of the main ones. So here, other finds that we support, we corroborate the transfer of Orexita and Aidoia to Schizonicini, and we also recover Hermionica, which is a monotypic genus that was previously positioned inside Dorinotini as strongly related to the tribe, which can provide further support for potentially bringing the species back into Dorinotini, as it was classified before the catalog in 1999. Then we also, given the knowing that our sampling is relatively modest to answering questions regarding whether these are monophyletic groups or not, here I just want to highlight some genera that were recovered as monophyletic, and they are Agriconota, Coptocycla, Dorinota, Basipronota, and Acrocassis. And we also can highlight the groups that were recovered as paraphyletic, Caridotella, Caridotis, Microctenochira, Cassidinae, and Plagiametrona. What is really not a big surprise for many of us. So based on this tree, we also did the divergence time estimation and biogeographic, historical biogeography reconstruction. We recovered the DAC model as the best, with the best likelihood. So basically members of the tribe expanded their ranges stepwise, often speciating after colonizing new regions. Based on our divergence time estimation, we recovered 44 million years as the crown age for Cassidini, and also the Neotropics was recovered as a major center of origin and dispersal. We also recover multiple recent dispersals events between 2 and 5 million years from the Neotropics to the Nearctic, likely enabled by the Isthmus of Panama. Important to note that these range gains are concentrated in nodes younger than 10 million years, aligning with the late Neogene biotic exchange. We also recovered a major split in the Cenozoic that gave rise to one lineage radiating in the Americas and another in the Afro-Eurasia, with likely route North Atlantic land bridge. But I must say that here our confidence intervals does not really align with this hypothesis, so we need a little bit of more investigation on the process that might have helped on this dispersal. And we also recovered dispersals from Africa to Madagascar that happened to a few endemic Malagasy lineages, likely result of one or two dispersal events from Africa, possibly occurring through the Mozambique Channel exposure that happened between the late Eocene and early Oligocene. We also recovered dispersals between Afrotropics and Indo-Malayan that were possibly enabled by the formation of the terrestrial connection via the Arabian Peninsula during the late Oligocene, which created a migration corridor between tropical East Africa and South Asia, allowing terrestrial organisms to move between lands. And in fact, the late Oligocene experienced relatively warm temperatures, and there were widespread tropical and subtropical biomes extending across East Africa, Arabia, and South Asia. We also recovered dispersal to Australasia, happened during the mid-Miocene, from possibly enabled by rainforest continuity across the Indonesian archipelago. And finally, many species originally lived across both tropical Indo-Malayan and Palearctic, but vicariance process likely led the split of these widespread populations into isolated groups through the geological uplift of the Himalayas, through the second phase of this geological uplift, or climatic cooling, So basically, we still have to make these inferences a little bit more robust. So the next steps of our study include investigating host plant associations as possible facilitators of dispersal and potentially explore co-dispersal and host tracking scenarios. And finally, all this really shows how the Bell Grant helped generating this baseline to explore morphology, host plant evolution, and it's been incredibly impactful to support. It really allowed the construction of a framework to study the subfamily and propel us into the genomic area, which was quite fundamental to a homoplastic group like Cassidines, and that is only known. There are many rare species only known from very few specimens. So I would like to offer my sincere thanks to the Coleoptera Society once again for supporting and promoting such initiatives, and I extend my deepest gratitude to the Bell family for their support and generosity for funding this project. Their commitment to coleopterology is truly commendable, and I'm honored to be selected as one of the recipients of their grant. So with that, I thank you once again, and I'm very happy to take any questions.